è riuscito, e quindi ora do la parola a Michael Pusch per chiudere questa giornata sui 50 anni della Società Italiana di Biofisica pure applicata, nel ricordo, direi piacevole e dovuto, ad Antonio Borsellino. Grazie Michael.
Okay, thank you, Alberto. I will switch to English, so or maybe Martin can also have some more insights here. It's the last talk. I hope you don't fall asleep. I try to be not too technical, and it's about endolysosomal anni transport from biophysics to human neurological disease, and this will be the main thing that this research is connected to disease. Some people will recognize this marine laboratory in Camogli, and I was unfortunately not able to work there because it was already closed, more or less. But it's a very nice place. And my PhD advisor, Walter Stümer, actually performed his PhD in this marine laboratory, so quite funny. And just a few general remarks, as many people have done this, what keeps us biophysicists off the street? And I think the most important thing is the curiosity about marvelous biological phenomena, like, for example, self-perception and intelligence, what is memory, or another very interesting thing is the extreme energy efficiency of biological systems. Like, for example, the brain has, I think, more computing power than a supercomputer, but it only consumes like 20 watts or so, in contrast to normal supercomputers. And for example, another things are the evolution, how these extremely complicated things like photosynthesis evolved. How could this happen? And I think since we are not creationists, there must be something how these things can be invented. And then we are maybe also possibly useful biophysicists. We can help in curing diseases. We can help in saving the environment. Maybe some people think we can help the industry, even though I think the industry should help themselves. Then just a few examples where physicists can help biologists to address biological questions. For example, we have seen many examples of this, like spectroscopy and photosynthesis, X-rays and electron microscopy for structures of biomolecules, then light microscopy for the cellular structures. And this is also, these are all developing methods. And bioelectricity, and this will be my topic today, where the electrophysiology can help. And I came in contact to the Italian biophysics, because Genoa has a long tradition in ion channel biophysics. And one main player was Franco Conti, who was here in Göttingen, where I was doing my undergraduate and my PhD work. With Walter Stümer, I did my PhD with Erwin Neher, who is not on the picture, and my diploma. And here they are meeting to work on a common project on the sodium channel. You have Shosaku Numa, who had cloned the sodium channel, and there's Bert Sackmann, still younger, and here's Keiji Umoto, who did all the work, actually. So this is like, I think it's around 86 or so. And then after having been in Genova for a year, I went back to Germany in the group of Thomas Jentsch, who had discovered a completely new gene family of anion transporting proteins, the so-called CLC family. And this world is small. Here you see my PhD advisor, Walter Stümer, and my postdoc advisor, Thomas Jentsch, at some Gordon conference in the United States. So it's a small field. Coming back to bioelectricity, one of the fundamental questions that was addressed by the biophysicists not only in Italy, but starting with Hodgkin and Huxley doing the voltage clamp, is what is the mechanism underlying the action potential? What are the ion channels responsible to generating these electrical signals that travel along distances and very, very fast? And the ion channels were eventually discovered, and now with the invention of molecular biology, having found the first ion channel genes of the action potentials, an incredible zoo of different ion channels had been discovered. There are hundreds of ion channels, potassium channels, chloride channels, sodium channels in the genome, and they are present in every cell, and in every membranes are present these ion channels. And one particular example that I have been studying in Thomas Jentsch's lab, it was the so-called double-barreled torpedo chloride channel. It's a chloride channel that is localized in the electric organ of the torpedo, and is one part of the mechanism to generate the high voltages. And it's a peculiar channel because here you see the current on the x-axis, and on the y-axis you have the time on the x-axis and the current on the y-axis. Somebody, I think Borsellino would have asked for that, I guess. And this was studied by, it was discovered by Chris Miller in the United States. And he came up with this double-barrel shotgun model, according to which the channel has two independent permeation pathways, as is seen here in these independent openings of two seemingly independent pores. And there's also a common gate that shuts off both pores simultaneously, as you can see here by the long closures here. And again, it's a small family here. Chris Miller was a good friend of Franco Conti as well, and he came to visit Camogli in 2004, and he gave a nice talk. And after cloning, after Thomas Jentsch had cloned this torpedo chloride channel, it was then relatively easy with molecular biology methods to find all human genes that are similar to the torpedo chloride channel. And I will particularly focus on one of these. And as I said, these ion channels or transporters are not only present in the plasma membrane, but they are also present in intracellular organelles. Like these CLC proteins here are found in endosomes and lysosomes. And in fact, the ion transport in all these intracellular organelles is as important as is the ion transport also in the plasma membrane. So they're important for the ion composition. They determine the membrane potential of these organelles. There are events of calcium release and so on and so on. This is still largely unexplored because it's much more difficult to access these intracellular organelles than the plasma membrane. So there's still a lot to discover about the biology of all of this. This I already said. And one interesting thing is that when the structure was solved first by X-ray crystallography, but then also by cryo-EM of various CLCs, this double-barrel shotgun model was completely confirmed. So you have it's a dimer, and you have two independent ion permeation pathways, one in each monomer. And then now the data I will talk about is a very small part. I will only mention this result. This was a very, very nice collaboration with a large number of people, which I only know very few of them personally. And the main players were Elizabeth Palmer, Emma Palmer, and Vera Karlscheuer, who are geneticists and who contacted me to study mutations that cause disease, mutations of the gene that encodes the CLC4, because I had expertise in CLC4. We started this collaboration, and this is the disease affects mostly children, so it's mostly a pediatric disease, but also some older people. And interestingly, CLC4 is X-linked, so more males are normally affected, but it's interesting that also females, heterozygous females, can have very severe disease, which is was difficult to explain, and we found a nice explanation for that. And here are the two geneticists, Emma Palmer from Sydney, Vera Karlscheuer from Berlin, together with a lot of doctors. We analyzed functionally in Genova more than 50 novel variants that have been discovered in the CLC4 gene. And the method is relatively simple. We produce the mutants in the laboratory, we generate RNA, inject the RNA into frog oocytes, and then can measure after a few days the channel in the membrane, either by electrode voltage clamp, or by patch clamp technique. So one example, I will provide you two examples. One is this mutation that initially looks like it has just less current. So it seems like a loss of function mutation. Here's where you see the wild type current. This is time. This is current at different voltages. Here you see the simulation protocol. You elicit, and these currents are elicited at rather positive voltages. And superficially, this just looks like less current. However, if one pushes the voltage to much more positive values, one will see, we discovered that at these positive voltages, this mutant actually recovers. It has almost the same current magnitude as the wild type, but what is changing is the voltage dependence. This mutant is much more rectifying, so it has a stronger voltage dependence. As you can see here, the ratio of mutant and wild type current is greatly diminished here. So this mutant affects the gating of the channel. It makes it much more difficult to open. And such an effect can be expected to be dominant in heterodimers. And this would explain the dominance, and that also female patients can have a phenotype because of this dominant effect. And in fact, the residue is located at the interface of the two subunits. Another example is another mutation. It's actually another disease with the there's another gene, very related CLC3. This also looks, again, it looks like having less current. Here you see the wild type current, the mutant current. But when one puts the extracellular solution acidic, one discovers that, at contrast to the wild type, which has no inward, no negative currents here, there is considerable negative currents here. They look small, but for these proteins, this is like a huge signal. So this is like a toxic gain of function effect that again explains a phenotype in females. In fact, this mutation is only found in females. For males, it would be probably disastrous. The fetus would not survive. Okay, this is just already the end, and this just want to say that this is the aspects we are considering. There are two faces of the same coin. On one hand, the human mutations actually guide the structure-function relationship. It reveals novel molecular mechanisms of protein function because it is those that cause the disease, that disrupt the protein in an interesting way. On the other hand, the biophysical analysis helps the patients clarifying, because when we clarify the pathogenicity, this can mean an end of a diagnostic odyssey for the families. When they know, okay, this mutation is indeed causing the disease, they have something in hand. It opens then for the families connection to recommendations for management of the disease with active patient support communities. It can help in reproductive counseling, and then it is also relevant for possible therapeutic strategies. And there are not only these neurodevelopmental diseases I talked about, there are many ion channel diseases, so-called channelopathies, involving, for example, voltage-gated sodium channels, calcium-activated chloride channels, another chloride channel, CFTR, voltage-gated potassium channels, and the CLC transporters I talked about. And there are many people at the Institute of Biophysics in Genova working on these diseases, and it's still expanding because with the next sequence generation technologies, more and more variants are found, and there is a pressing need to do this functional analysis. And I would like to thank you for your patience.
Grazie Michael, veramente grazie molte per questa chiusura e per questa chiusura di giornata. Ma prima di chiudere